Escherichia coli, also known as E. coli. In this lecture, we will cover all aspects of the E. coli. So let's get started. E. coli is a gram-negative, facultative, anaerobic, and rod-shaped bacterium. Facultative anaerobic means that it can survive in both aerobic and anaerobic conditions, means in the presence or in the absence of the oxygen. It is the normal flora of the human intestine, and it has two strains. One is non-pathogenic and other is pathogenic that cause severe diseases in the human body. If we talk about the classification of the E. coli, so domain of the E. coli is bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Enterobacteriales, family Enterobacteriaceae, genus Escherichia, and species is the Escherichia coli. Morphology, it's a rod-shaped bacteria, that is bacilli. Size of E. coli is 1 to 3 micrometer long and 0.4 to 0.7 micrometer wide. Gram reactions, it will look pink after the gram staining. So it is actually a gram-negative bacteria because gram-negative bacteria will look pink after gram staining. Motility, most strains are motile. Why? Peritrichus flagella. Capsule, some strains have polysaccharide capsule, also known as K antigens. So that is the important thing in the pathogenicity. And spore formation, this is the non-spore forming bacteria. Cultural characteristics, media. E. coli grows well on nutrient agar, McConkey agar, and EMB agar. These, these are the most important media on which E. coli grows. On the McConkey agar, it forms pink colonies due to lactose fermentation. EMB agar, metallic green sheen, especially E. coli. So that is the important thing. You can see in the figure, left side, you will see the McConkey agar and you will see the pink colonies formation. And on the right side, you will see the EMB agar. You will see the green, metallic green sheen. That is the characteristic of E. coli. And the optimal temperature for its growth is 37 degrees Celsius. Biochemical characteristics, catalase positive, because E. coli produce the catalase enzyme, and through the biochemical reactions, we can detect the enzymes produced by the bacteria in our case, that is E. coli. So E. coli produce catalase and we can detect it through biochemical reaction. So it is the catalase positive. Oxidase negative, indole positive, methyl red MR positive, Vogus Proskauer VP negative, citrate utilization negative. TSI, triple sugar iron slant. In this test, you will see the two portion. Bottom portion will be the butt and upper portion will be the slant. In both cases, you will see the yellow color formation. Yellow color forms due to the productions of the acid. So in that case, slant and the butt, bottom portion, butt and the upper slant will both turns into yellow. It means in both bottom and the upper portion, you will see the productions of the acid. And along with that, you will also see the bubble formations in the test tube. So in that case, gas produce also. But there is no H2S formation because when those bacteria that produce the H2S will turns the color into black. So this is the TSI test for E. coli. Antigenic structures, O antigen somatic lipopolysaccharide, H antigen flagellar antigen, K antigen capsular antigen, and F antigen fimbrial antigen. So different components of the bacteria will behave as antigen. OHKF antigen in case of E. coli. Next is the types of the pathogenic E. coli. That is the most important thing. Diarrheal E. coli that cause the diarrhea. First is the enterotoxigenic E. coli. These are the different strains of the bacteria that cause different diseases in our body. ETEC that cause traveler's diarrhea. Enteropathogenic E. coli, EPEC, infantile diarrhea cause. Enterohemorrhagic E. coli, EHEC, bloody diarrhea, HUS, hemolytic uremic syndrome. Specifically, what type of strain? It causes O15787. Enteroinvasive E. coli, dysentery-like illness, enteroaggregative E. coli, persistent diarrhea, diffusely adherent E. coli causes the watery diarrhea. Next is the extraintestinal pathogenic E. coli, uropathogenic E. coli causes urinary tract infection, neonatal meningitis, E. coli, neonatal meningitis, septicemia-associated E. coli causes the bloodstream infections. These are the important strands of the bacteria that cause different diseases in our body. Virulence factors, fimbriae, that helps in the attachment to the uroepithelial cells. Exotoxin, there are three types of the exotoxin produced by E. coli. Heat labile increases the cyclic AMP. This is the mechanism of the heat labile toxin. Heat stable increases cyclic GMP. This is again the different mechanism as compared to the heat labile. Shiga-like toxin inhibits protein synthesis. These are the important exotoxin. Capsule protects from phagocytosis, and LPS, endotoxin, causes inflammation and septic shock. Clinical manifestation, gastrointestinal infection causes watery and bloody diarrhea, abdominal cramps, vomiting, and hemolytic uremic syndrome, specifically caused with the EHEC strains of the E. coli. Urinary tract infection, dysuria, difficulty in urination, frequently urination and fever, and pyelonephritis, infections of the kidney. So this is the symptoms related to the UTI. Other infections, neonatal meningitis, pneumonia in hospitalized patients, sepsis, especially in the immunocompromised individual. Laboratory diagnosis is specimen collection, stool, urine, blood, CSF, depending upon the type of the infections. Microscopic gram-negative rods and culture. On the McConkey agar, you will see the pink colonies, and on the EMP agar, you will see the green metallic sheen. Biochemical test in DOL positive, MR positive, VP negative, and lactose fermentation. Serology, especially for EHEC O15787. Molecular test PCR for septic toxin and ELISA for Shiga toxins. Treatment for the UTI and sepsis, the important antibiotics: ciprofloxacin, ceftriaxone, nitrofurantoin, and TMP-SMX, trimethoprim sulfamethoxazole. For the meningitis neonatal, ampicillin and gentamicin. Antibiotic not recommended in EHEC may worsen HUA. That is the important thing in the HUS, hemolytic uremic syndrome. Sportive therapy, rehydration, oral, or it can be IV line, electrolyte corrections, and dialysis in HUS. Prevention, proper hand hygiene, safe drinking water, proper food handling and cooking, avoid raw and undercooked beef, pasteurized milk, and vaccine under development for a specific strain like ETEC. Public health importance, major cause of childhood diarrhea in developing countries, commonest cause of UTI in females because due to the anatomy of the female. Important nosocomial pathogen, risk of foodborne outbreaks. Summary is gram stain, gram negative rods, motility, motile, but there are some exceptions. Lactose fermentation, positive. Virulence factor, it produces toxin, fimbriae, capsule, LPS, endotoxin. Diseases, diarrhea, UTI, sepsis, and meningitis. These are the important diseases caused by the E. coli. Diagnosis, culture, biochemistry, and serology and PCR. Treatment, antibiotic, strain specific, depend upon the strains of the E. coli. That will be decided after the, obviously, after the culture of the uh, bacteria. Next, prevention, hygiene, safe food and water. So this is all about the E. coli. If you still have any question, you may ask in the comment section. Thank you so much.